Hello, I'm Fairlie Reese from the Mortazavi Lab at UC Irvine. The title of my poster is Swan, a Python library for the analysis and visualization of long-read transcriptomes. To summarize this work, we felt there was a need for a tool to perform downstream analysis after calling isoforms from long-read RNA sequencing data. Additionally, we needed a better way to visualize said isoforms to fully understand their splicing complexity. When applying Swan, my Python library, to encode PacBio HepG2 and HFFC6 long-read sequencing data, we found around 4,000 differentially expressed genes and 5,000 differentially expressed transcripts. Additionally, Swan found 279 isoform switching genes where there is a transcript called as differentially expressed when the parent gene is not. Swan also discovers 1,021 reproducible novel exon skipping events and 73 novel intron retention events. As you know, genes often contain multiple transcript isoforms such as these ones. Short-read sequencing data relies on error-prone transcript assembly that cannot unambiguously resolve the entire transcript's sequence. Long reads circumvent the issue of assembly by sequencing through the entire transcript, which is important because differences in transcript structure can lead to functional impacts such as coding sequence changes or regulatory differences in the UTRs. The data used for this pilot study consists of mapped data from 2 replicates of HepG2 and 3 replicates of HFFC6 sequenced on the PacBio Sequel1 from the ENCODE portal that my lab generated. Some metadata about the runs is shown here, and you can look at it further on the poster if you wish. I then processed the data using the ENCODE long-read RNA processing pipeline that we developed in our lab, called Talon. Talon assigns each long read a transcript identity. Novel transcript models are then filtered for those that are highly reproducible and do not exhibit internal priming. Finally, Talon generates a transcriptome for each dataset and quantifies transcripts. Here are some transcripts that Talon calls from the data shown on the genome browser that belong to the gene CSTB. As you can see, in this visualization, it's difficult to tell what the difference is between any two transcript models. Knowing that these visualizations lack in informative ability motivated me to develop this software. Swan is a Python library that uses graphical representation of transcript models to store, compare, and visualize transcriptomes. Swan represents unique splice sites as nodes, colored by their usage in the transcript. Transcription start sites are blue, transcription end sites are orange, and nodes that are internal to the transcript are yellow. Representing individual splice sites as nodes rather than entire exons as nodes allows for more nuanced examination of events such as alternative 3 and 5' splice site usage. Introns and exons are represented as edges between splice sites. Introns are pink edges and exons are green ones. As seen here, when using Swan, you can add transcript models from an annotation or from a dataset in the form of a GTF or a Talon database, which is the output from the pipeline presented earlier. Adding an annotation allows for assessment of novelty for transcripts, splice sites, and splice junctions that are added from datasets. As seen in the diagram, nodes and edges that are only seen in the datasets can be plotted differently than those annotated. Novel nodes are outlined, and novel edges are dashed. Abundance information can also be incorporated when adding transcript models from a dataset. To cement how Swan represents transcript models in its representation, called a SwanGraph, here is an example of what 2 traditionally represented transcripts from the same gene might look like in the SwanGraph. Each unique splice site in the transcript models is labeled with the corresponding node in its SwanGraph. Each unique exon and intron is represented as a connection between splice sites. Exons are colored green and introns are colored pink. This is an example of a real gene summary graph that Swan can produce. A gene summary graph shows all the possible splicing combinations in a specific gene, in this case GLTPD2. This plot also highlights unannotated splice sites as outlined nodes and unannotated introns and exons as dashed edges. In this gene, Swan identified a novel intron retention event, a novel exon skipping event, and a novel alternative 3' splice site event, Each of these can be seen in the dashed edges in the plot. Swan can also make gene reports that display each transcript model from a given gene alongside information such as expression level, transcript novelty, and differential expression results. This gene is interesting because not only does it contain a novel exon skipping event in the 4th transcript, but Swan also called it as an isoform switching gene, as you can see by the expression differences for the last 2 models. The last 2 models are completely absent in the HepG2 data, but are expressed in HFFC6. If we zoom in on the models and display the browser versions of them as well, which Swan is also capable of rendering, you can clearly see the novel exon skipping event in the 4th transcript model here. In the future, I'm interested in both applying Swan to new datasets, such as the other ones that my lab has generated for ENCODE, and to long-read single-cell RNA sequencing data. On the development side of things, I'm excited to finally get input from users on what features or improvements they'd like to see in the software, as well as add some new analysis tools such as examining transcript starts and ends or predicting ORFs and NMD transcript status. And of course, I'd like to thank my lab, especially Dana and Gabby, who I've worked with very closely on long reads for the past 2 years, as well as my funding sources. I'd also like to mention that you can download Swan on GitHub at the link shown here. And I thank you for your time.